Okay. Thank you so much, Masood. It's a real pleasure. I know your work. I know the Enigma Consortium and the papers, and it's really amazing. And as Masood was saying, I'm talking from Argentina. So just to take some interest on how rhythms research is here, you can go to a bookstore and look for a book on circadian rhythms. And this is the situation. You might find this book, an old book by a French guy called Alain Rimberg, on biological rhythms. But the interesting thing is, who are our neighbors, right? Who do we share our bookshelves with? And here I can show you this. As you can see, we share bookshelves with aloe vera, miraculous plant, predictive astrology, Ernesto Che Guevara, life and work, of course, a very well-known chronobiologist, and this one up here, whom I love, which is the big book of cooking, right? So that is where Argentinian chronobiology is standing, at least from the viewpoint of a bookkeeper, right? So let's get out of there and go back to real science and real life. And I'm not going to talk only about animals because I'm really switching for the last 5 years or something to human studies, but I'll try to make sense on how to complement both, right? Animal and human studies. So I'll tell you about an animal model we've been working with for the last, I don't know, 15 years or so, what happens with this circadian desynchronization model, and then just to give you a taste of what we are working on, what happens with human synchronization, human circadian rhythm synchronization. You are very well aware about circadian rhythms, but the real question is about time and time in biology. And time actually spans lots of scales from milliseconds, like sound localization or electric organs in fish, for example, have a period of milliseconds. And then you go to seconds, which is something very, very well known in cognitive neuroscience, what happens in the second range. And then we go to the two main scales of time in biology and in neuroscience. They are so important that they have their own names, right? We have interval timing that has to do with the seconds to minutes time estimation and subjective time scale. And we do work in that scale a lot, but I'm not going to talk about this. And of course, the diurnal cycle, the 24-hour cycle, which is the circadian rhythm scale, which is the one we work most on. But that actually spans another question, because are all of those rhythms related or completely independent? So one of the things that we've been doing in the lab was to try to find interactions between interval timing, circadian timing, and even larger periodicities like seasonal timing. Again, I won't go into this, but it's something that we need to think about. How do different times interact in the brain and in our behavior, right? For example, one of the things we've been very interested in is looking at circadian rhythms. Let's see if I can go with a laser. Yeah, here we go. How circadian rhythms interact with second to minute timings. And it's very different. If you want to estimate 30 seconds during the day or during the night, it will be very different. And again, it will be very different throughout the seasons. So there is clearly a very strict interaction among different time scales. And again, we are all neuroscientists, and we usually take into account time into a few of the variables that we usually consider, like if we do measurements of electric potentials on the brain, or of course reaction time, or of course memory, conditioning, attention, aging has to do with time. But timing for neuroscience and biology is much more than that. And there are a few questions that we usually don't ask ourselves when we design and analyze an experiment, right? Like, what is the brain doing with time? What parts of the brain deal specifically with time, right? So we have very different places in the brain, like we have clocks in the cerebellum that have to do with the millisecond timing and have to do with the precise movements that span a millisecond range. Then we have the interval timing circuits, like, for example, the corticostriatal circuits that have to do with dopamine and GABA, and they actually command interval timing. And then we have the circadian timing mechanism with a master clock in the SEN, in the suprachiasmatic nucleus of the hypothalamus, that interacts with many other parts of the brain, and also has a privileged dialogue with the pineal gland, which acts as a diurnal and seasonal signal for the rest of the body. This is sustained in genetic clockwork that we know very well in different models, from Drosophila to humans, that have to do with a transcription, translation feedback loop of specific genes that, whenever they are mutated, we have phenotypes that affect circadian rhythms. So I was telling you that there's a few questions that we usually don't ask ourselves when we design an experiment. Like the most easy one: do you take into account the time of the day when you're performing your experiment? The usual answer is no. We usually do experiments during the day, and we use nocturnal rodents as our main model. So most of what we know about behavior and physiology has to do with the opposite time in which we should be measuring things. What happens with the environment? Do we keep a controlled cyclic environment? Like if we do an experiment with mice or with rats, or of course with primates or humans, do we know what time it is in terms of the light-dark cycle? Do we know what's going on in terms of temperature, in terms of changing cages and so forth? Do we also consider sleep as one of our main variables? Well, we do, because this is the ENIGMA consortium, and you do measure sleep. But what about feeding time? That we know, we've known this for about, I don't know, 10 to 15 years, that feeding time also affects circadian rhythms. And then let's suppose we're measuring, I don't know, reaction time or memory or whatever experimental procedure. What about if that procedure itself is affecting the temporal structure of your subjects? And the other main question, what we need to ask ourselves, is what's going on with light, right? Light has many effects, not only a vision effect. There are the so-called non-phodic effects of light, or non-vision effects of light, that affects behavior, mood, physiology. And this is something that we need to stress. Like just one very simple example about this. Let's take the ICU, right? The intensive care unit. It's not a place where you have days and nights, right? Because you don't turn lights off during the night, and there's a lot of noise during the night as well. And you probably know that if you just turn lights off in the ICU, the patients will be better 20% faster than if they don't have nights. So light has to be our main companion, our main hero for designing experiments. Another thing that we need to take into account, as I told you, we know quite a lot about the so-called clock genes that are involved in the feedback loop of the circadian clock. Well, they are not only involved in circadian rhythms. Most of clock genes are pleiotropic, and you'll find that many of them have to do with behavior, with mood, and even with mood-related disorders. So again, this is something I would like to call out for any of you that are working on mental health or neurology or so forth. Also consider that there might be changes in clock genes that affect what you're measuring, right? Now let's talk about the circadian system itself. The usual model of the circadian system is a very simple one, a linear one, that has to do with a Zeitgeber, which is a time giver, and most of you are in Germany, so I don't have to translate this. The main Zeitgeber is, of course, the light-dark cycle, not the only one, but the one we know most. And the light-dark cycle or other Zeitgebers entrain a circadian clock, and the master clock, of course, is located in the SCN. And the SCN, by different coupling mechanisms, including neuron and humoral coupling mechanisms, tells the body what time it is, right? So this is called an eskinogram because the first scientist that proposed this very simple scheme was Arnold Esking, a wonderful guy in the circadian field. But it's too simplistic, right? So we started looking at different arrows that put together these mechanisms. For example, the Zeitgeber might not go through the circadian clock, but go directly into the circadian rhythm. That's called a masking phenomenon. Also, rhythms might feed back into the clock, right? And actually, behavior might actually control the effect of masking of the Zeitgeber itself. So it's not an easy model, actually. And it's starting to look quite much more into what's called the Ouroboros. It's a mythical creature, the dragon that eats its own tail. So right now, it's even much more complicated because, as you, of course, all know, there is not just a central circadian clock, but there are peripheral circadian clocks all over the body, mainly with local influence. That this means that these feedback loops have to be repeated in most organs of the body and in most cells of the body. So it's really a complicated system. And there is someone that thought about such a complicated system much before we chronobiologists did, which is this guy. This is the main Argentinian writer. You might know him. It's called Jorge Luis Borges, incredible writer. And in one of his short stories, he thought about an inflated series of time, which consists in convergent and divergent times. In some of these parallel times, time can be forked, can be broken off. In some of these times, you exist and not I. In others, I and not you. In others, both of us. This is a story called The Garden of the Forking Paths, right? So was he talking about circadian desynchronization itself? I think so. So how can we translate this metaphor by Borges into our circadian model? Actually, this is circadian disruption, right? In some specific conditions, we'll get disrupted rhythms because there will be dysregulation of a part of our circadian model, which could be from the Zeitgeber to the clock, from the clock to the rhythms, or all together. And we have many models of circadian disruptions, mainly in animal models, but of course we also have them in humans, right? Going from continuous light to shift work to jet lag and so forth. The circadian disruptions can be measured behaviorally. That's kind of the easiest way to do, right? I reckon you're all very familiar with actograms. If not, just let me go very quickly through this. An actogram depicts locomotor activity or can be adapted for any other variable in which you see all of the consecutive days plotted against 24 or 48 hours, right? So here what I'm showing, and this is real data, I have to say, is what happens if you place light at night in a nocturnal rodent or continuous light, or you do a forced desynchrony protocol, which means that the light cannot be predicted, right? It will change throughout the different cycles. And what you can see as a phenotype is that not only locomotor activity will change, but also feeding activity, the transcription of clock genes in the SCN and in the liver, bona fide markers of the clock like melatonin and so forth. Also, one of the main phenotypes that you get after a circadian disruption is metabolism, which is the same that happens in humans. Once you have a very good model of circadian disruption, one of the first phenotypes you're going to see is changes in metabolism, like changes in weight, changes in insulin sensitivity, and so forth. In our hands, we've been working with a very specific model of chronic circadian disruption, which we could call chronic jet lag, because it's the equivalent of somebody traveling through 6 time zones every 2 days, right? So we advance the light-dark cycle 6 hours every 2 days. I know it's a very hard protocol. It's not something that resembles something that happens in humans, but it's very good to see potential phenotypes. And we started looking at this and we found very strange things under this protocol. This is a double plotted actogram of a mouse under an light dark 12-12 cycle. And this is under the 6-2, 6 hour advance every 2 days. And what you can see is that the clock is broken, right? Remember Borges, remember the forking paths. We keep circa 24-hour component, which is this one I'm signaling here. It's actually closer to 25, and very short component here as well, right? So this is the analysis of that of the model. Here you can see what happens from an LD cycle through the disruption paradigm back to DD to see what happens to the clock. And what you can see is that during the disruption paradigm, you'll see these two circadian components I was telling you about. This is Fourier parallelogram analysis. You see the 21-hour component and something closer between 24 and 25, right? This is specific for chronic phase advances, and it doesn't happen if you delay the light-dark cycle 6 hours every 2 hours. What you'll see is that the clock tends to follow the delay and computes this as a 27-hour day, right? So we are focusing here on the chronic phase advance paradigm. And well, we are not only looking at behavior, of course, we want to take a look at what's going on with the main circadian clock genes in the SEN. And as you can see here under LD cycles, there is a very clear day-night variation, very well known for PER1 or BMAL1, which is in the opposite phase. And this is completely lost under the chronic jet lag conditions or the circadian disruption condition. We have other genes as well, right? And as I told you, one of the phenotypes we were quite interested in was metabolism. Here you can see what happens with the weight after about 1 month under the chronic circadian disruption paradigm. Under LD, you will get, of course, a little increase in weight, but it's much significantly increased under the chronic advance paradigm, and this does not happen under the chronic delay paradigm. Of course, you might ask, do they eat the same? Yes, they did. They eat exactly the same, and in the bottom panels, you'll see the amount of food that they are consuming. I won't go into this a lot, but we started looking at different ways to counteract this increasing weight. One of those was providing the animals with a running wheel. So with voluntary activity, you don't get the increase in weight. Another way is time-restricted feeding, which we've been exploring for quite a while. And another one is group house the animals, right? If you group house them, they won't get an increase in weight. Again, we wanted to go further into metabolism, and as you can see, there is a clear change in the diurnal variation in TGs, triglycerides, in liver weight, in adipocytes, and in fat in different organs. And why is this happening? Because under chronic phase advances, the animals get a lot of food during their subjective day, right? So what we found here in red is superimposed the feeding activity to the black locomotor activity. And as you can see here in panel B, you'll get much more red during the day. And that's the food that increases weight, right? So what kind of obesity model is this? Well, it has to do with an impairment in glucose metabolism. Again, there's a very clear rhythm in glucose utilization under LD conditions, and this rhythm, this actually day-night variation, is lost under the chronic jet lag protocol. We started looking at microbiosis here, at microbiota. This is kind of expensive because we have to sequence all of the bacteria, and well, we cannot afford this, but at least we know that again there are some very clear and well-known diurnal rhythms in the proportion of the different bacteria that are in the gut, and this variation is lost under the chronic phase advance cycle. We wanted to know if this was trophic or hyperblastic. And clearly what we find is not an increase in the number of adipocytes in the liver and around the gut, but it's an increase in the size of adipocytes. So this is good news, actually, because if this is the case, we can counteract this with diet and other variations, right? As I told you, one of the ways to go back to the original weight was to restrict feeding to the night. This is a nocturnal animal. These are mice. So when you do restricted feeding, as you can see, weight goes back to the control values, both at day 11 and day 30 of the protocol, right? As I told you again, getting a running wheel will counteract the effects on locomotor activity and also on weight, right? Oh, sorry, this. Okay. Then we started looking at other phenotypes of desynchronization, like, for example, mood-related phenotypes, and started looking at motivation, right, in which you measure the amount of lever presses for a reward, a food reward, or other kinds of reward. And clearly, there is an anhedonic phenotype for the desynchronized mice. We looked at anxiety and depression-like behaviors, and just to summarize, the animals do have higher levels of anxiety-like behavior and depression-like behavior. So circadian desynchronization is clearly affecting not only the circadian clock, but some outputs in terms of mood, right? And what might be one of the most spectacular effects of this model has to do with how the body copes with experimental tumorigenesis. We implanted melanoma cells, 3LL cells, in mice, and as you can see, in black is the control under LD conditions. The tumor will grow in volume, but in gray here is the tumor growth under the chronic circadian desynchronization condition. And as you can see, it's a much more significant and much faster growth, which results in a lower survival rate and in a lower latency until you can measure the existence of the tumor here. So actually, circadian disruption is affecting the clock gene expression in the SCN and in the periphery of the animals which carry the tumors. And not only clock genes, but also we've been looking quite a lot into immune-related genes to look at what's going on in the pathways that allow the body to defend itself from the tumor, right? In particular, we looked for cell cycle genes. And again, what we found is in general, and to summarize a lot of results, is that there is a very clear circadian rhythm in cell cycle genes, which tends to be lost under circadian disruption conditions. So this was published a couple of years ago, and our latest result in terms of phenotypes of circadian disruption has to do with another tumor model, which is B16 cells, which are metastatic cells. You implant them subcutaneously, but they induce metastasis in the lung. And as you can see here, under chronic jet lag, chronic circadian disruption conditions, the induction of metastasis is significantly higher than under LD conditions, right? And actually, we get many more metastatic loci and much bigger as well. So there's something going on with circadian disruption. We are looking at the pathways that have to do with metastasis right now as we speak, that are exaggerated under chronic circadian disruption conditions. So this was the main message I wanted to tell you about some of our animal results, and specifically what we've been doing with circadian desynchronization models in mice. Again, it's a chronic phase advance protocol, 6-hour phase advance every 2 days. And we get not only locomotor activity and, well, I didn't show it, temperature disruption, but also metabolic phenotypes, behavioral consequences, and tumoral consequences in this mouse model. So again, you could say, well, but that's weird, right? That's a model that does not exist in the world. In the world, we are subjected to very clear days and nights. And right now, if anything, we would have the problem of light at night more than circadian phase advances or delays. So the question here could be, are we prepared for a world that does not exist anymore? A world in which there are very clear days and nights, there are very clear seasons? Well, according to some places, this world does exist, right? So many years ago, as soon as I graduated as a biologist, I went to look for circadian rhythms in places where the world is as it was in pre-industrial times, like, for example, Antarctica, right? And I measured rhythms in penguins many years ago. But the other condition is a place in Argentina, are places in Argentina in which there are very big seasonal variations. And in some cases, you do not get the artificial Zeitgebers. So we measured rhythms in populations in Antarctica, right? And right now we are measuring rhythms and sleep in populations in rural communities in Argentina that, when we started this, had no access to electricity, right? We found 2 populations in the north of Argentina which we could compare, because one of those had access to electricity, one did not, right? It was the perfect comparison because they were genetically very similar, culturally very similar, but the main difference was the access to electricity. So let me show you about how to work this, right? Well, if you want to look at environmental modulators of sleep and circadian rhythms, you could do lab experiments. They're not real, right? You could analyze big data, which we all do, of course, but we don't have any control. What if we could compare communities in which the possible modulators of sleep and circadian rhythms are differentially present, right? And that's where many rural communities in South America come into place, like in Brazil or in Argentina. I'm mentioning Brazil because some colleagues of us have been very successful in studying some communities in Brazil, right? There are quite a few field studies in communities with different levels of industrialization, right? We started, as you can see, in 2004 in Patagonia. Many people in Brazil have done this, both also in Africa and a few others around the world. In our case, we found these Tobaquon people in northeast Argentina, which are hunter-gatherers. They are very, very similar in terms, as I told you, genetic and cultural. But we found an urban population of these people and a rural population of these people in which we could compare the effect of electric light, right? And this was published, this is a collaboration with my friend and colleague Horacio de la Iglesia at the University of Washington in Seattle, which we were schoolmates many years ago. And what we found is that in the absence of electricity, you'll get one more hour of sleep, and you get much more seasonal variation in sleep than in the urban population. And this happens both in the summer and in the winter, right? Um, but this has been going on for a few years now, right? 12 to 15 years. So things have changed, right? When we started this, the two populations had very different conditions in terms of electricity. Right now, the rural population also has access to electric light. They also have TVs, and some people even have their cell phones. So what had happened in the meantime? Well, this is a satellite image of our two populations. The one down here is the urban population. As you can see, throughout the years, it has more light, but here is the rural population. No light at night when we started, some light at night a few years ago, and quite a lot of light at night right now. So this is the perfect condition in which to study the effect of light on these populations. And as you can see here, and we've just submitted this, we hope it goes well, the onset and offset of sleep in these populations has changed. So in summary, they have lost about 1 hour of sleep throughout this 10 to 12 years of research. So this is a real life situation in which we can see, as it evolves, the effect of environmental light, artificial light, sorry, in sleep, right? So second take-home message, we were able to measure let's say, ancestral conditions of sleep without access to electricity. And very recently, and this is something we just finished analyzing, what happens when you introduce electric light in these populations. And as I told you, there is a very clear delay in sleep onset due to the light. And this results in about a 1 hour reduction in sleep duration in this population. Finally, right, how do we measure desynchronization in real-life urban scenarios? Some time ago, we introduced this notion of sleep capital, right? It has to do with how sleep, and specifically synchronization, has some economic, productivity, and of course, health consequences across the lifespan. And in this equation of sleep capital, we kind of proposed quite a few inputs for the notion of sleep capital, which have to do with what time it is in terms of time zones, in terms of access to electricity, in terms of chronotyping, in terms of schedules of people, and outputs of sleep capitals in terms of what is the benefit for health, for mood, for economy, for productivity of sleep capital, right? The problem here for measuring, and this is something that you know much more than us here, is of course the data for measuring all of this. And where do we get the data from? Well, National Health Service, mental health service that usually include some sleep questions, specific sleep surveys, and something that is quite comparable across different countries, it is time use surveys. In our hands, at least for South America, they're all useless, right? Because they're not reliable, or they are not repeated periodically, or many other reasons, right? So what do, right? We have to go and do our own surveys. Like, for example, a very large survey we are conducting in Argentina in terms of sleep duration. We now have like 25,000 subjects for this. And the conclusions are what you would expect, which is we are not getting the required 7 hours of sleep during the night. As you can see, the average is about 6.5 on weekdays. And of course, we increase that during weekends, which results in a very large social jet lag figure, right? So once we decided that we could not rely on surveys, we want to look for data wherever it is. So we want to look for proxies of circadian rhythms, right? Something that is legally authorized to act on behalf of another party. So we looked at many different circadian proxies, like, for example, chess playing activity. You know, people play on the internet throughout the day. There are really crazy people that play at 3 a.m. or 4 a.m. or whatever, social networks, cell phone use, transportation data, and so forth. We did quite a few studies on Twitter activity, and we found that it's a very good proxy for social jet lag, right? As you can see here, there's a very clear phase delay in Twitter activity on the weekends, right? And well, we did many other things, but this is not what I wanted to tell you. This is our latest idea. What if we could use any kind of interaction with smartphones as a proxy of circadian rhythms? So we've been doing this with Android because it's the main type of cell phone that is used in Argentina. And let me just go to the results here. This is interactions with a cell phone of a single participant throughout quite a few, almost a year. Yeah, and as you can see, it looks like an actogram. It looks like if you were measuring activity with any kind of wearable, right? And we can even look at social jet lag with Android activity, with cell phone activity. And here you can see how we can superimpose activity recordings in black with cell phone interactions in red. Of course, this is noisy because interactions might mean anything, but this is a very good window into activity with which we we will be able to measure desynchronization paradigms as the one I've been talking to you before, right? Final take-home message, right? If we want to measure synchronization and desynchronization, it's very easy to do that in animal models. It's not that easy to do in modern societies. In our hands, it's hard to do this with survey data. So we are looking at circadian proxies as an approximation, right? So we tend to imagine our cities in space, but we actually live in time, not only in space, right? So that means that everything that I've been telling you about, we need to translate this into public policies. Public policies that have to do with time zones. In your case, in Europe, most of you are in Germany or in Europe, it has to do with DST, the discussion into changing time throughout the year. The discussion regarding light at night, and more recently, light during the day. The discussion regarding the circadian hospital, at what time do things happen in health systems, the discussion that has to do with school timing, specifically for teenagers, discussions that have to do with shift work or working across the clock. So we should consider ourselves satisfied with our research if at some point it serves as evidence for public policies. But again, we need to continue doing research. That's why I think we still need to continue sharing data between animal models and human subjects, because it's the only way we'll understand our main question, which is, of course, time. But time waits for no one, and it's about time that I end this talk, and I'm very happy to answer questions.